everyone. My name is Ishitha, and today I'll be presenting on Alpers-Huttenlocher syndrome by going beyond the mutation to explore its global impact and the urgent need for research. But before we dive into the science, a little bit about me. I'm a rising junior at Ravenwood High School, located in Brentwood, Tennessee. I am the founder and co-president of Cultural Connect, as well as a HOSA two-time ILC qualifier and attendee. I am also involved in Red Cross, Student Council, Science National Honor Society, Rho Kappa Honor Society, Mu Alpha Theta Honor Society, Model United Nations, and Youth in Government. I hope to pursue a career in pediatrics or neuroscience. So, what exactly is Alpers-Huttenlocher syndrome? Commonly known as AHS, Alpers-Huttenlocher syndrome is a severe neurodegenerative disorder that typically appears in children between the ages 2 and 4. It is caused by mutations in the POLG gene, which affects the brain's gray matter, an area that cannot regenerate itself. Children with AHS often experience severe seizures, developmental regression, liver dysfunction, and problems with movement and vision. This disease gradually strips children of their abilities and ultimately their lives. This MRI scan shows just how severe the neurological damage can be. The red arrows indicate brain tissue loss, also called atrophy, while the yellow circles highlight areas of hyperintensivity, which signal inflammation or fluid backup. These changes are focused in the occipital lobe and the angular gyrus, regions essential for vision and language. They reflect how deeply AHS affects brain structure. But to truly understand why this happens, we have to look even deeper at the gene behind it all. And that gene is the POLG gene, which stands for DNA polymerase gamma. It's responsible for replicating and repairing mitochondrial DNA, which your cells rely on to produce ATP. When POLG mutates, it causes a depletion of mitochondrial DNA, and the whole process breaks down. Mitochondria can't work properly, and the cells, especially in the brain and liver, begin to fail. AHS happens when a child inherits two defective copies of this gene, one from each parent. You can see here how both parents carry the mutation. This is because it's an autosomal recessive inheritance, and because of this, each child has a 25% chance of inheriting this mutation. Families often don't know they carry it until their child shows the symptoms, but by then, the damage is already underway. Despite AHS affecting approximately 1 in 100,000 individuals globally, it's part of a much larger challenge. Many rare diseases go under-recognized and overlooked, creating major obstacles for the people who have them. According to the World Health Organization, over 300 million people worldwide live with rare diseases. And with over 7,000 known rare diseases, about 95% of them have no improved treatment. This, in fact, includes Alpers-Huttenlocher syndrome. Care is limited to managing symptoms and trying to improve the patient's quality of life. Because it's so uncommon, Alpers-Huttenlocher syndrome often gets overlooked in healthcare funding, policy decisions, and medical training. With thousands of other rare conditions facing the same fate, I chose to focus on AHS to understand its cellular impact and raise awareness for diseases the world often ignores. So, to explore AHS at a molecular level, I focused on a key mutation in the POLG gene, A467T. The guiding question was, how does this mutation impact mitochondrial DNA integrity in cells? My teammate Reese McKenzie and I hypothesized that bovine cells with the A467T mutation would show altered banding patterns during gel electrophoresis, indicating disrupted mitochondrial replication and stability. Our goal is to understand how this mutation affects the mitochondria, guiding future therapies. To answer our research question, we designed a 6-step experimental plan. The first step is to culture wild-type bovine cells in a DMEM media. The A467T mutations will then be introduced into selected cells to create a genetically modified group. Next, the mitochondria will be isolated by lysing cells and centrifugation. Mitochondrial DNA will then be extracted and amplified using PCR with M13 primers. Results will finally be put into a gel electrophoresis by size to observe changes in the DNA before finally using a nanodrop spectrophotometer to quantify the DNA and the integrity of the DNA. If our results show distinct bending patterns and reduced mtDNA in mutated cells, it will confirm how the A467 mutation disrupts mitochondrial function, the core of Alpers-Huttenlocher syndrome. But understanding this isn't enough. The next step is treatment. CRISPR-Cas9 offers one path forward. It edits nuclear DNA and shows a real promise for creating and correcting POOL mutations. Now, there is also a new mitochondrial base editing tool named DDCBE, a tool developed by the Broad Institute. It edits mitochondrial DNA directly without damaging nearby genes. This could change how we treat mitochondrial diseases altogether. Beyond that, future research will use patient-derived stem cells to better model AHS and test treatments in the lab. One day, we might even insert healthy synthetic mitochondrial DNA to fix damaged cells at their source. This work isn't just about one disease. It's a starting point, building targeted therapies for rare disorders that have waited too long in the dark. Every step forward deepens our understanding and brings us closer to real solutions. I would like to thank my research partner, Reese McKenzie, for her hard work and collaboration throughout this project, and a thank you to my teacher, Danielle Drumright, for her inspiration, guidance, and support, as well as a thank you to the Global Health Leadership Conference for this opportunity. And lastly, a thank you to you, the audience, for your time and interest in my project. So I leave you with the question. If we have the tools, the science, and the chance to rewrite our future of rare disease care, what's stopping us? Here are the references I used throughout my research on this project, and here is my contact information. Thank you.